Proteins and amino acids. Proteins are compounds consisting of carbon, hydrogen, oxygen, nitrogen, and a little sulfur. The monomers of proteins are amino acids. There are 20 different common amino acids. The word peptide refers to proteins. A tripeptide is made up of 3 amino acids. A polypeptide is a chain of many amino acids. A protein is made up of one or more chains of amino acids that have folded into a functional shape. Proteins carry out the work in an organism. They're the machines, the enzymes, the transporters, the receivers of messages, and more. They also form some important structures. When you look at another person's body, most of what you see are their structural proteins. At the cellular level, structural proteins help cells move and retain their shapes, and help things move inside the cell. Amino acids are made up of an amino group, a carboxyl group, and a side chain, all attached to a central CH. Each unique side chain determines the chemical properties of its amino acid. An amino group is polar, basic, and can accept a hydrogen ion from solution. It's found in bases and ammonia. A carboxyl group is polar, acidic, and can donate a hydrogen ion to solution. It's found in organic acids, like acetic acid. A sulfhydryl group binds to other sulfhydryls to form SS. It's found in proteins. For the shown small biological molecule, a tiny piece of a protein, highlight the nitrogen carbon-carbon backbone of the molecule and highlight the functional groups. Here is the nitrogen carbon-carbon backbone of the molecule. Here is the amino group, the carbonyl group, the hydroxyl group, and the carboxyl group. The chemical bond that is formed between amino acids is called a peptide bond. The bond is between the amino group of one amino acid and the carboxyl group of the other. Like bonds between saccharides and nucleotides, they're formed by dehydration synthesis. This shows formation of a dipeptide by a dehydration synthesis reaction. Trace the backbone through the dipeptide. The carbon backbones of proteins have this pattern. There are 20 amino acids from which all proteins are comprised. Amino acid side chains are categorized according to their chemical properties. They may be nonpolar, polar, positively charged, or negatively charged. Amino acid side chains contain familiar chemical groups, which determines their behavior. It is not necessary to memorize any chemical structures or names of amino acids for the AP Biology exam. 2 amino acids, aspartic acid and glutamic acid, are negatively charged at neutral pH. Their side chains behave like acids. They have carboxyl groups that can donate a hydrogen ion to the solution. These negatively charged side chains are attracted to positively charged side chains. 3 amino acids, lysine, arginine, and histidine, are positively charged at neutral pH. Their side chains behave like bases. They have amino groups that can accept a hydrogen ion from the solution. These positively charged side chains are attracted to negatively charged side chains. Polar amino acids have either hydroxyl groups or amino and carbonyl groups. These side chains are attracted to water. They can also participate in hydrogen bonds with water or with other polar molecules. The nonpolar side chains are composed of carbon and hydrogen, or sulfur and hydrogen. These side chains are hydrophobic. They repel water and are attracted to each other or to lipids. Cysteine is unique because it has a terminal sulfhydryl group, SH. Analyze this molecule. Trace the backbone of the molecule. How many amino acids are shown? What are their names? What is the general name for this type of molecule? How many peptide bonds are shown? Highlight them. Circle the terminal amino group. Circle the terminal carboxyl group. Draw rectangles around the R groups. Here in yellow, we have the backbone of the molecule. There are 4 amino acids, leucine, glycine, serine, and alanine. This molecule is a tetrapeptide, which is a chain of 4 amino acids. The peptide bonds are shown with red arrows. Here is the terminal amino group, here is the terminal carboxyl group, and here are the R groups.